Hello, everyone. Welcome to the Columbine Health System Center for Healthy Aging's Healthy Aging Speaker Series. My name is Allie, and I'm the admin at the center, and I'll be your host for today's presentation with Dr. Chris Bell, which is titled Cannabis, Sativa L, and Exercise, to THC or not to CBD. Now, what was the question? The Center for Healthy Aging at CSU serves as a catalyst for interdisciplinary research, evidence-based outreach, and education about healthy aging. So we appreciate you joining us to learn about the great research happening in the aging field. If you'd like to participate in future speaker series, feel free to email me. I'll put my email in the chat. A few housekeeping items before I introduce our presenter. Please be sure to mute your microphones so that any background noise can be reduced. There should be time after the presentation for Q&A, and you're welcome to either unmute yourself and ask your question or type it in the chat. Also, we are recording this talk, so you can view it again or forward the link to friends, family, and colleagues. It will also be on the center's website along with the rest of the series. And now I'd like to introduce Dr. Bell. Chris Bell was born while still very young in the damp recesses of Northern England. Worn down both physically and spiritually by the gray skies and relentless horizontal rain, he fled to North America to pursue sunshine and higher learning. At the University of Western Ontario, he spent his days pumping older adults full of electricity and his nights learning about hockey and other barbaric Canadian pastimes. In 1999, just prior to the falsely advertised Y2K mediated end of the world, he relocated to the University of Colorado Boulder for postdoctoral training and high altitude shenanigans. Following the signing of an undisclosed transfer fee, Bell relocated to Colorado State University in 2005, where he has continued to be a considerable disappointment. He currently lives in a dream, but shares a home in Fort Collins with his imaginary friends and a long-suffering wife, Karen, from who he steals most of his best ideas. Thank you, Dr. Bell. Go ahead and start when you're ready.
Thank you, Ali, for that introduction. Hi, everyone. This is obviously a one-way conversation, so I'm going to assume that at least a third of you said yes, some of you said not yet, and the remainder of you are only just realizing that I actually asked a question. Let's get the dull stuff out of the way first. Oh, actually, the first dull thing is getting the screen to advance. Here we go. Several of the original pieces of data I'll be sharing with you today were collected as part of studies that were funded by industrial entities. So I would like you to please bear that in mind as you interpret my interpretation of the studies. Here's what I'm going to be serving. This will be a 40 to 45 minute, 5-course menu of plant-based food for thought. Call me predictable, but I'm going to start at the top and work down. I'm going to begin by introducing the plant in question, Cannabis sativa L. This is quite a complicated plant. The last time I counted, there were over 545 different chemicals and compounds inside this plant. Arguably, the chemical that is the most notorious is, of course, delta-9 tetrahydrocannabinol, THC, notorious, of course, because of its psychoactive properties. Not too far behind is cannabidiol, abbreviated as CBD. This is non-psychoactive and is thought to have lots of medicinal properties. More on those medicinal properties a little later. Also worthy of mention is hemp. Hemp are products that are derived from the cannabis plant that contain less than 0.3% THC. Now, those of you reaching for your calculators, this means that you need to ingest about 1.6, 1.7 grams of hemp in order to receive 5 milligrams of THC. That's actually enough. It'll take the sting out of the day. Other chemicals worthy of mention, we have the up-and-coming delta-8 THC. This is also psychoactive, not quite as strong or even as prevalent within the plant as Delta 9. However, with a little bit of genetic manipulation and some crossbreeding, I suspect we're going to be hearing a lot more about Delta 8 over the next few years. Other chemicals worthy of a nod, there are lots of other cannabinoids. There are also terpenoids. These are the chemicals that give the plant its distinct aroma and flavor, and then several flavonoids and alkaloids. By way of lexicon, by way of vocabulary, so that we're all talking about the same thing today, when you hear me refer to cannabis, I will be referring to the plant. When I talk about marijuana, I will be talking about Delta 9 THC, Delta 9 tetrahydrocannabinol. Cannabidiol will be abbreviated to CBD, and we can assume, unless otherwise stated, that that is free from THC. And finally, if you ever hear me refer to funky broccoli, it means I've become delirious and we should probably end the conversation. So with that amuse-bouche out of the way, let's start with our appetizer. Let's talk a little bit about some physiology. And to illustrate the physiology, I've got a health and exercise science student from Colorado State University. Good grief, the student is naked. Here, let me provide some clothing. There you go, buddy, that'll make you feel a little better. As humans, we have an endocannabinoid system. We are able to generate chemicals on our insides that have a lot in common with many of the chemicals that are found in the cannabis plant. There is a party in each one of us just raging to get out. Lots of different endocannabinoids. The two about which we know the most are anandamide and 2-arachidonylglycerol. These are thought to evoke their effects through cannabinoid receptors. There are two, and these have been creatively named CB1 and CB2. And if I risk the modesty of our health and exercise science student, then in red, I can show you the approximate anatomical distribution of these receptors. Originally thought to be predominantly located in the central nervous system, we now know that they are highly prevalent also within the periphery. Similarly, in blue, these are the CB2 receptors, and again, these are fairly prevalent throughout both central and peripheral locations. It would be so convenient if this was the end of the story. However, as often the case with physiology, it's a little bit more complicated. It turns out that the endocannabinoids are also able to evoke their effects through multiple channels and also several different types of receptor. And if I were to color these receptors in white, then you can see that they are ubiquitous. Wherever we look for these channels and these receptors, we tend to find them. If you're not someone who thinks about receptors on a daily basis, I think it's worthwhile reviewing a little bit of receptor physiology. Here is a receptor sitting on the surface of a cell membrane. An agonist is something that will bind to the receptor, and upon binding, it will initiate a cascade of intracellular signaling events that will evoke a physiological response that we can measure. An antagonist is something that will competitively bind with the receptor. It then occupies the binding site such that future agonists can no longer interact with the receptor. An antagonist is known as a blocker, and a beta blocker is a common example with which people are quite familiar. An inverse agonist is something that behaves like an agonist, except when it binds to the receptor, it actually inhibits all of the downstream signaling pathways, so we don't see this measurable physiological response. And finally, and arguably the most fun to say, we have a negative allosteric modulator. This is something that upon binding to the receptor. The receptor and some of its downstream signaling messengers will undergo a conformational change. It will change shape, and the receptor is no longer available for stimulation by an agonist. So why am I telling you all this? The reason is that if we now consider cannabidiol, CBD, I can tell you that, at least with respect to the CB1 and the CB2 receptors, CBD has been shown to behave like an antagonist, an inverse agonist, and a negative allosteric modulator. How can it react to all three? How can it behave like all three? It really comes down to the experiment that you're using to describe the actions. And by that, I mean, what tissue are you investigating, whether the tissue is within the body or whether it's been separated from the body. And then, of course, whether the tissue is in man or beast. If we consider THC, tetrahydrocannabinol, this is shown to be a partial agonist of CB1 and CB2. And again, really the definitive outcome depends on your experimental design. That was quite a quick review of receptor physiology. If you have a thirst for more, then I can recommend this review paper. It's very thorough. It came out quite recently, and most importantly, it is open access. You don't need any library or journal subscriptions. Anyone with an internet connection should be able to read this paper. So, with that physiology in mind, the big question then is, what can CBD and/or THC do for you? And indeed, if we were to believe the marketing, certainly the marketing for over-the-counter products, an easier question to answer might be, what can't CBD and THC do for you? Some of the claims made by these commercially available products have been outrageous, and the science doesn't always support these claims. Unfortunately, some of the science is a little bit contradictory. There are several studies that show a beneficial effect of CBD with regards to one outcome, while other studies may show a negative effect or no effect at all. And the reason for that is the perfect segue to our next topic, that of pharmacokinetics. I believe that much of the discrepancy within the science can actually be attributed to a failure to consider pharmacokinetics. So let me tell you what I mean. If you were to ingest some cannabidiol, and then we were to look at the concentration of cannabidiol in your circulation, in your blood, then we might get a figure that looks something like this. So on the x-axis, we have time. On the y-axis, we have the concentration of cannabidiol in your blood. The curve is going to resemble something along the lines of this shape. We'll see an increase in cannabidiol as it leaves your stomach and enters the blood. It will eventually attain a maximal concentration, and from there it will slowly decline. This curve can be described mathematically. There are several key features of the curve. The first is the maximal circulating concentration. That is known as the Cmax. Often more important than the Cmax is the time it takes to get to that maximal concentration. And that is known as the Tmax. These two variables, of course, refer to a very specific point in the curve. And other people like to consider the entire curve all in one variable. So for that, we can calculate the area beneath the curve, and that is thought to represent the total exposure to the drug. There are lots of other variables that are associated with pharmacokinetics. These are just the three that I'm going to be talking about today. We, in my lab, have actually compared and contrasted the pharmacokinetics of 5 different commercially available cannabidiol preparations. These preparations differ in a number of ways. Some of them are water soluble, some of them are lipid soluble, and they differ by the presence and absence of various excipients and emulsifiers, things that will hold the cannabidiol together and help with its transport out of the stomach and into the circulation. You can see from this figure that formulation 707 appears to be the superior product. It has the highest Cmax. It has a very comparable and fast Tmax compared to the other formulations, and it also appears to have the greatest area under the curve, therefore the greatest exposure to the cannabidiol. Something else to point out, you can see based on those error bars, there is considerable variability in the concentration of cannabidiol within individual participants. To explain that, we have considered size and shape. And for this, I'm going to again borrow a health and exercise science student. We don't all look like health and exercise science students. Of course, people come in all different shapes and sizes. We have used very unstandardized, highly variable participants, and to them we have administered a very standardized dose of cannabidiol. It stands to reason then, based on these different shapes and sizes, that the resultant concentrations, the absorption and the distribution of the cannabidiol will be very different between participants. We were able to quantify body size and body composition using something called dual energy X-ray absorbiometry, or DEXA. This is essentially a whole body X-ray that tells us how much fat and how much muscle we have and where it's all located. Looking at our individual body composition variables, we were unable to find any single variable that was able to predict with any degree of consistency any of the pharmacokinetics of the cannabidiol. So while intuitively appealing, it appears that shape and size are perhaps not that important when it comes to looking at the pharmacokinetics of cannabidiol. Something else we standardized in our experiment was how much food people had eaten prior to our experiment. We had every single person in the studies fast for 12 hours prior to the start of the studies. If you're considering cannabidiol for exercise, it's unlikely that you're not going to eat for 12 hours before you ingest your cannabidiol. So in this study, we've actually looked at what are the influences of food on the pharmacokinetics. Here in the broken purple line, we are looking at the pharmacokinetics of cannabidiol when it is co-ingested with a liquid mixed macronutrient meal, so a meal that contains fat, carbohydrates, and protein, and the total caloric content was equivalent to approximately 40% of resting metabolic rate. What you should see compared to the solid line is that the Tmax has been lengthened, the Cmax has been increased, and the overall area under the curve has been increased by the food. Something else to consider. Our data were preliminary data, so they are still in review, but we're not the first to demonstrate the influence of food. This study from a couple of years ago was looking at the pharmacokinetics of Epidiolex. Epidiolex represents pharmaceutical grade cannabidiol. This is highly regulated, approved by the FDA for the treatment of seizures. In this study, you can see that not only have they looked at the influence of food on the pharmacokinetics, but they've compared different types of food. And what you should see here in the squares, the green squares, is that food that contains the highest amount of fat and has the highest caloric load leads to the greatest lengthening of the Tmax and the greatest increase of the Cmax in the area under the curve. We have also looked at the pharmacokinetics of 5 different commercially available edible marijuana preparations. This is actually quite an important question for some obvious reasons. If you consider the pharmacokinetics of smoking marijuana, they are considerably faster than if you are using edible marijuana. Imagine the scenario. 5 minutes after ingesting your edible marijuana product, you don't notice any change in your psychoactive state. So then you decide to take 1 or maybe 2 more. Then 20 minutes later, you've suddenly realized that your neighbor's unicorn has been stealing the toppings from your pizza. In this study, you can see that the Ripple gummies, illustrated in red, appear to be the superior product. They have the highest Cmax. They have a very fast, easily amongst the fastest of the Cmaxes, excuse me, and they have the greatest area under the curve. One thing to consider, though, when we are looking at commercially available CBD and THC is that we shouldn't always believe what is written on the label. This study, and there are several other studies like this, have actually compared different over-the-counter products. On the x-axis of each of these figures are commercially available products. On the y-axis, they've measured the cannabidiol content of each of the products and expressed it as a percentage of what's written on the label. If your favorite product is in the green zone, then you're getting exactly what you paid for, within 10%. If your product is in the yellow zone, then you're actually getting more than what you bargained for. And the CBD in this area is greater than 110% of what is listed. This, in some instances, might be good. You're getting a bargain. In other instances, it might actually increase your chance of overdose. Finally, if your favorite product is in this lower pink zone, then you're getting ripped off. You're getting less than 90% of what is advertised on the label. In fact, if you look at the lower panel, you'll see that there are at least half a dozen products that don't actually contain a measurable amount of CBD. More serious than that, there are some brands of CBD that advertise themselves as being THC free, but you can actually measure THC within the products. If you're an athlete, this can be a disaster. It might mean you lose your place in the Olympics. If you are somebody who lives in a state where THC is still illegal, then you might lose your job. You might even spend some jail time. So grim. Don't always believe what's written on the label. Right. To summarize this little section, if you are considering ingesting cannabidiol or THC for your exercise, remember that you need to consider the timing of the effects. The pharmacokinetics between different products are very, very different. While intuitively appealing, it doesn't appear that size and shape of our bodies are an important determinant of the pharmacokinetics. On the other hand, food will lengthen the Tmax, increase the Cmax, and increase the area under the curve. And finally, don't believe what's written on the label. Okay, now we get to the main event, the main course. Let's consider the interaction of CBD and THC with respect to exercise. Athletes looking for gold, should they consider the cannabinoid or should they cannavoid? Before we get into the details, let's first consider, is this an important question? And I would argue, yes, it is. Based on this meta-analysis from 2020, the responses to questionnaires were collected from over 46,000 athletes of various different ages and abilities. From those 46,000, almost 25% of the respondents reported using a cannabis product within the previous 12 months. So that's almost 1 in 4 people who exercise are using cannabis products. We don't even know if this is a good thing or a bad thing. So let's take a look at some studies. To my knowledge, there are 3 studies that have given cannabidiol in an acute setting and looked at the exercise response. This study is the most recent study and arguably the strongest of the three. Here's the schematic of the experimental design. People reported to the lab first thing in the morning. They were either administered a placebo or cannabidiol with food. They were given a brief period of time to absorb the cannabidiol, and then they performed 2 bouts of exercise. The first was very, very standard. It was a 1-hour run at a moderate exercise intensity. The other, after a brief break, involved maximal exercise to exhaustion. Things to emphasize: the cannabidiol dose was 300 milligrams. That's quite a large dose. And the exercise testing took place 90 to 200 minutes after the ingestion of the cannabidiol. The reason that I think this study is amongst the strongest is because they were the only study that actually measured the circulating cannabidiol after ingestion. The light green lines that you see here on the screen, they represent the individual responses. The dark green line represents the group average. If we superimpose the exercise testing over the top of this figure, then you can see that for all but one of the participants, Cmax had not been attained. That is, at the end of the exercise trials, CBD concentration was still increasing. They hadn't yet reached their maximal concentration. The implications of this are a little hard to interpret. One reason that it may not be that important is if you look to the y-axis, you'll see that these concentrations are actually quite high, considerably higher than the concentrations that we were reporting in our pharmacokinetics trial. And this is probably due to the fact that they've given 10 times the dose. We gave 30, they've given 300. Now let's see what they found. How did it influence exercise? Before I explain these data to you, I want to point out that the authors were very quick to emphasize that this study was a pilot study. They weren't necessarily looking for statistically significant differences. Rather, they were interested in effect sizes, i.e., to what degree of certainty can they claim that cannabidiol may have influenced a physiological response to exercise. So with that thought in mind, I can tell you that the only variable that appears to have been influenced by the cannabidiol was the oxygen consumption, the oxygen cost of the exercise. It appeared to have been increased during the 1-hour run. That would suggest that the runners with the cannabidiol have somehow become less efficient. Their running economy may have suffered. None of the other variables, the rate of production of carbon dioxide, the respiratory exchange ratio, none of those things were influenced. And on the lower panel, none of the parameters known to be important for determining ventilation, none of those were influenced either. On the second slide, still looking at the 1-hour run, the only variable that appears to have been influenced by the cannabidiol was blood lactate concentration, and that was only at one specific time point at 40 minutes, with the idea that the blood lactate was greater with cannabidiol. There didn't appear to be any influence of the cannabidiol with respect to heart rate, blood glucose, or the rate of perceived exertion. You'll remember in this study, they also finished with some maximal exercise. If we look at the maximal data now, I can show you that the only variables to be influenced by the cannabidiol were maximal oxygen uptake. Maximal oxygen uptake is thought to be an indicator of aerobic capacity, how successful you'll be when you complete an endurance event such as marathons and half marathons. This was increased with the cannabidiol, but the increase is quite small. We also see an increase in the maximal respiratory exchange ratio. The relevance of that is also a little unclear. The other two studies that I mentioned looked at cannabidiol more in the context of recovery from exercise. Both studies involved research participants completing a brutal resistance training session, lifting lots of big heavy weights, with the idea being that they were trying to evoke muscle damage. Following those brutal workouts, the participants were given cannabidiol, in this study a 60 milligram dose, and then over the next 3 days, blood was sampled to measure circulating markers of muscle damage. The only beneficial effects we see of the CBD were on the third day. We see that on the third day, there's a lower concentration of creatine kinase and a lower concentration of myoglobin. Creatine kinase and myoglobin are things that exist on the inside of our skeletal muscle. If we can measure them in blood, it usually means that we have somehow damaged the muscle, we've ruptured some membranes, and these chemicals that should be on the inside have leaked out. So this isn't a good thing. But the idea that cannabidiol has reduced these markers after 3 days is potentially beneficial. If we look down here in the lower left, we can see that this lower muscle damage also was accompanied by an improvement in the 1 repetition maximum of the back squat. It's a very small improvement, but nevertheless, it is an improvement. We didn't see any effect at all on a dynamic type of activity, such as the counter movement jump. The third study didn't actually have any physiological measurements. All of their data was based on the perception of the research participants. What we're looking at here on the y-axis is perceived soreness. And what we can see is that over the 3 days following that brutal workout, there was an increase in the perception of muscle soreness. They all reported having very, very sore, achy muscles. And you can see that the cannabidiol did not influence this response. So far, cannabidiol's influence on exercise and exercise recovery is a little underwhelming. Perhaps more thought-provoking are some of these earlier studies, not looking at cannabidiol, but actually looking at the cannabinoid receptor. This study was performed in humans. What we're looking at here is 48 hours after a bout of resistance exercise, the expression of the CB1 receptor has been decreased. This led the authors to speculate that perhaps the CB1 receptor is somehow involved in the molecular pathway by which we grow our muscles. Let me explain. In response to a mechanical stimulus, we will see increased anabolic signaling through the mTOR pathway. If you're not familiar with this pathway, this essentially is just a series of chemical messengers that are telling our muscles it's time to grow. Growing muscles is very, very expensive from a caloric perspective. So sometimes we have a governor, something that might limit that response. The authors speculated that this may be the CB1 receptor. And if we were to inhibit the CB1 receptor, maybe we'd all get bigger muscles. This idea is actually consistent with these more recent observations. If we compare two populations that are typically different in terms of the amount of muscle mass we have, such as a group of young, healthy men in their early 20s compared with a group of older men over the age of 65, you can see that the expression of the CB1 receptor is greater in the older men. So that is consistent with the previous ideas from the other study. It gets a little bit more complicated when we look to see what happens if we put those men on a 12-week resistance exercise program. We actually see an increase in the expression of both of the receptors. So how that fits with the hypothesis is a little less clear. But if we think for a moment about the receptor slide that I showed you, and I remember that I said that cannabidiol might inhibit these receptors because the cannabidiol behaves as an antagonist or an inverse agonist or a negative allosteric inhibitor, then what might be worth looking at, worth considering, is whether or not ingesting CBD prior to resistance exercise, if that actually gives you a bigger skeletal muscle response. To my knowledge, only one study has begun to investigate this. This study was following a single bout of resistance exercise, and the study was completed in rats. What we're looking at here on these two panels that you see at the bottom, we're just looking at circulating concentrations and expressions within the muscle of some of the chemical messengers that are instructing our muscles to grow. If you focus on the white bars, so the control is before resistance exercise, stimulation is after resistance exercise, you can see that there's an increase in these chemical messengers. That's good. If we now look at what happens when we administer CBD and the rats do the same exercise, you see exactly the same response. So the cannabidiol didn't appear to influence the anabolic signaling. So if I can summarize this cannabidiol section, then the results are fairly underwhelming. It appears that acute cannabidiol, while it may evoke modest effects on the physiological response to exercise, it doesn't appear to be appreciably ergogenic. It's not going to improve your exercise performance. Which then, of course, brings us to the final point, the dessert, if you like, in our meal today, and that is the influence of exercise and tetrahydrocannabinol, the interaction between the two. Before I begin, let me recommend this review to you. This is a recent review, again, very, very thorough, and again, open access. All you'll need is an internet connection if you want to read more on this topic. Here's a fun fact about review papers. There are more review papers addressing the topic of exercise and THC than there are studies that have actually examined the influence of exercise and THC. In fact, to my knowledge, there are only 3 studies that have looked at the acute effects of THC on exercise performance in healthy people. More noteworthy, those studies were performed in the 70s and in the 80s. Thus, their idea of good research might be a little different then as to what we think it is now. They were limited by their apparatus, by their technology. Even the marijuana was different. We know over the last 10, 15 years, the potency of THC inside cannabis has grown tremendously. Heck, they couldn't even spell it. This study was looking at the effects of marijuana on maximal exercise performance. Here's the study design in the lower panel, and this gets at a little bit what I was alluding to earlier about potentially poor research design. The study was simply measuring exercise performance after 10 minutes of smoking a marijuana cigarette or after 10 minutes of doing nothing. So straight away, we have a confounding variable. Inhalation of any combustible material is going to increase your exposure to hydrocarbons, to carbon monoxide, things that might limit your ability to transport oxygen. Let's take a look at the data. C represents the control condition. M represents marijuana. If we start at the left, we can see that there was no influence of marijuana on maximal oxygen uptake, no influence on the maximal production of carbon dioxide, no influence on the maximal ventilation, and no influence on maximal heart rate. The first indication that something may have gone awry within marijuana was a decrease in the time to exhaustion. The participants stopped the experiment early because they fatigued earlier and were unable to continue. This was about a minute less in the marijuana condition, and this was the equivalent of approximately 16 watts on the cycle odometer. This isn't a huge decrease, but it is a decrease. If we look at the continual data rather than just the maximal data, Then on the y-axis, we're looking at the percentage of the maximum effort. And then on the x and y, on the two y-axes, excuse me, we're looking at oxygen consumption and carbon dioxide production. You can see that in the end stages of exercise, the intensities that are probably greater than the lactate or the ventilatory threshold, we see greater oxygen uptake and greater carbon dioxide production with the marijuana. This is probably reflective of decreased efficiency, so not a good thing. And that brings us to where we are today. I am very excited to be able to share with you some preliminary data, some very preliminary data. I can't emphasize enough how preliminary these data are. Some might consider these data to be premature. We've completed testing on 4 people. These may be the only 4 people that respond like this. Our intention is to increase the study numbers till we get to somewhere between 10 or 15. Let me show you our study. We have looked at the influence of an edible marijuana product. We have looked at the influence of Ripple gummies. You'll remember the Ripple gummies from our earlier pharmacokinetic experiment. We've used Ripple gummies because we're very familiar with their pharmacokinetics. We looked initially at the responses to standardized cycle ogometer exercise. If we focus on the top left, I can tell you that in 3 of the 4 participants, oxygen uptake was increased with the marijuana, suggesting a decrease in efficiency. In the lower left, if we look at respiratory exchange ratio, we can see that in 3 of the 4 participants, this was increased. This could be interpreted as an increased reliance on carbohydrate or a decrease in the oxidation of fat, or it might simply be a reflection of the increase in ventilation, which we observed in 4 of the 4 participants. Finally, I can tell you that with the edible marijuana product, we also saw an increase in heart rate during exercise for all 4 of the participants. We also included a performance measure. This is a very common test used with cyclists. This is the functional threshold power test. This refers to the maximal mean power that you can sustain on a cycle ogometer for 60 minutes. 60 minutes is quite a long time. And of course, the pharmacokinetics and the circulating concentrations are going to change during that time. So we shortened the test to 20 minutes. This is quite a common compromise, and then adjusted the data mathematically to express what the power would be over an hour. The broken lines represent individual responses. The heavy black line is the group average. We see, just looking at the group average, a 1 watt difference. So certainly nothing to write home about. Finally, we wrapped up the day with a very, very tortuous and difficult sprint to fatigue test. You have to cycle as fast as you can until you can no longer cycle. This test is so difficult, only 3 of the participants were able to complete it. Again, individual responses in the broken line. The dark line represents the group averages. And based on these 3 observations, nothing again too compelling with respect to anaerobic performance. In summary then, our very tentative conclusion so far: acute THC may evoke modest effects on the physiological response to exercise, but it does not appear to be appreciably ergogenic. If we consider THC in the context of other ergogenic aids, other things that are known to improve your performance, such as caffeine or sodium bicarbonate, or even dietary nitrate and beet juice, the THC's effect is very, very underwhelming. In fact, I would say that for athletes who are considering using the green in order to obtain their gold, they may be better off eating their vegetables. And amongst those vegetables, I would include the funky broccoli, which of course means it must be time for me to stop talking. But before I do, I need to make some acknowledgements. Listed here on the left are some of the students who are currently members of our lab and former students. These, of course, are all the students that contributed to the data collection. My lab group tell me that they are the hottest thing in science right now, and they've submitted this thermal image as evidence of that. Those of you that know Taylor, he's missing from the picture because he was too cool to show up. Also on the right, I must acknowledge contributions from my faculty collaborators, Dr. Dooley from the Analytical Toxicology Lab, Tiffany Weir from Nutrition, and then our pharmaceuticals expert, Dr. Akagi. Finally, I must acknowledge our funding sources. And my very last slide, I promise, we are still conducting some cannabidiol and THC research, and we are in need of participants. If you would like to get involved or even learn a little bit more about the study, please email the lab at the email address that you see at the bottom of the screen. And with that, I'll shut up and try and answer some of your questions.
Thank you so much, Dr. Bell. Apologies, my internet was unstable for a moment and I thought you had frozen. I'll start with a question we actually got beforehand. I think I may have the beginning of arthritis in my hands. Will topical products help ingested? Is it anti-inflammatory?
So cannabidiol is thought to be anti-inflammatory, but if we look at the FDA approved uses of cannabidiol, really there's only one, and that's for the treatment of seizures. While it may have some benefits, the science for its use with arthritis is a little bit questionable. It's not entirely consistent. So you could try it, and if it works for you, fantastic, but just know that it may not.
Thank you. Next question was asked during, while you were covering the results of the 300 milligram CBD study. Does this imply a difference in metabolic substrate, more carb than fat?
Yes, an increase in respiratory exchange ratio is usually indicative of greater reliance on carbohydrates, so burning more carbs than fat. Unfortunately, respiratory exchange ratio is also influenced by ventilation, and a fairly well-established effect of THC and cannabidiol is an increase in ventilation. So it's tough to say if it is changing the fuel that you're using or if it's just an increase in ventilation.
Thank you. Are you aware of any research looking at chronic marijuana smoking on the subject's mesenchymal stem cell proliferation / pro-regenerative effects? In cigarette smoking, there is strong evidence that chronic cigarette smoking is highly detrimental to MSC function.
So the simple answer to that is no. I am not aware of any research in that regard. Sorry, I can't help.
Are your N=4 results adjusted for DLCO?
DLCO. So you're going to have to help me. What's DLCO?
Diffusing powerful. I'm sorry. DLCO. Diffusing power for carbon monoxide. What would the quality of gas exchange your lungs are doing?
I'm not quite sure I understand your question. Did we adjust for carbon monoxide? Is that what you asked?
No. Are their lungs healthy and of equivalent sizes?
Got it. So first of all, this was edible marijuana. So the lung sizes with respect to the THC ingestion, it wouldn't be influenced by the lungs. With respect to the research participants, then they were young, healthy people that all completed an exercise stress test and undergone a physiological examination. While they may not be exactly similar between participants, i.e. subject 1 may look a little different to subject 4, the key comparison here is we're comparing the same person to a control condition, i.e. that person exercising after a placebo and that person exercising after the ingested marijuana. So that's the key comparison. So I don't think we actually need to make that adjustment that you're referring to.
Is there an analgesic effect of THC? And if so, how could that affect the results, especially of your maximum power data?
There is some evidence, but again, it's not consistent, but there is some evidence that there is an analgesic effect. It is thought to help with either a decrease in pain or increase your tolerance. And for sure, yeah, that may be one of the mechanisms. In this first study, this updated study, we're really just taking a look to see, does the THC influence the exercise performance? If we find that it does, then obviously the future studies need to be taken into account some of the mechanisms by which it influences the exercise performance. And the analgesic properties are certainly a great thing to consider as we explain our findings.